this video, I'm going to teach you the cell and the membrane transport. Okay, so let me get the relevant PowerPoint. All right. So this chapter, we talk about human cell, obviously. Cell, which is already we learned, the fundamental unit of life. So what you are going to expect in this chapter, this is what you are going to expect. You have to learn what is a cell. Is there a different type of cells? So you need to mention that. And then you're going to study the structure and the function of the cell, which you are also learning in the for your laboratory classes. So you have to know the nucleus, the plasma membrane, and all the organelles. So if you really think about a cell is normally like when we study it, we say it's like a oval shape, but that is not case. That is not the case for every single cell that exists in human body. Different cell has different shape, but we are going to study, for example, using this kind of structure. Okay, so in a cell, there are three major aspects. Okay, I'm trying to explaining you first, like what do you expect from this chapter? Then we're going to go detail. Okay, so this is plasma membrane, which surrounds the cell, because animal cell, we do not have cell wall like plant. They have additional outside this, but we have this plasma membrane, which is normally called plasma membrane or cell membrane. So we need to know the property of the cell membrane, and there is a part of this chapter which discusses the transportation across that membrane, meaning how ions and molecules of various sizes and various property can go through them, like whether in to out or from outside to the cell to the inside. So that's you're going to study. And then inside all these things together we call cytoplasm. In the cytoplasm we have the liquid part, which is cytosol, and the fluid part is cytosol, and the rest, these structures are called organelles. The word organelles means tiny organs. So these are literally organs of a cell. So then in this particular PowerPoint presentation, I put a slide down in the middle about all the organelles, like starting with, like, let's say, rough endoplasmic reticulum, ribosome, smooth endoplasmic reticulum, Golgi apparatus, mitochondria, lysosome, peroxisome, cilia and flagella, cytoskeleton, nucleus, all these things is listed. So what are you going to know? You have to know what they look like and what they do. So mitochondria, what they look like. So you should be able to identify the mitochondria. Not identification required for the lecture class, but you should be able to know how they look like. So this is a mitochondria and what their function is. So that's pretty much of the chapter. So the first part, we're going to focus on the plasma membrane and the transport. And then second part, we're going to focus on all the other organelles and their structure and function. So it's a very straightforward chapter, I would say. Simple, comparatively speaking, but it requires a lot of study. Okay, so let's go back to our first slide. So Robert Hooke, when he was discovering, working with a microscope, he was looking at a piece of slice of a quark, and he saw those millions of tiny, like a unit, those are, looks like a beehives. So they call it, he called them cell, like a cell, like of a prison, meaning it is surrounded by wall everywhere. So that's become the name of the cells. Okay, so now for animal and plants, for every living organisms on earth, the fundamental structure is the cell, the fundamental unit of life. That is why it's called cell, because remember in the first chapter we learned that atoms and molecules makes the organelles. Organelles makes a cell. So cell is the fundamental unit of life. Although atoms and molecules are the smaller than cell, why we cannot call them? Because atoms and molecules cannot, like they don't have all the properties of a life has. For example, like they can't reproduce, okay? But the cell can. So cell can do the work that as a human, as an organism, you can do. Okay. So cell can reproduce, cell has the metabolism, cell can undergo growth and differentiation, cell can adapt, cell can be organized, and etc. So all the characteristics that is good for a human body, it's also good for a cell. That's why we are going to consider cell as the fundamental unit or the functional unit of life. There are two types of cell, not in human body, altogether we call eukaryotic and prokaryotic cell. The carry-on, the word came from the nucleus. So eukaryotic means good nucleus. EU means good nucleus. So eukaryotic means the cell has a nucleus which is membrane bound. That means nucleus itself has its own membrane. This is the picture. So nucleus has its own membrane. That's a nuclear membrane. So this nucleus has its own membrane, and they have also other membrane-bound organelles, that is eukaryotes. Prokaryotic cells, they do not have membrane-bound nucleus, and they also do not have any other membrane-bound organelles. In prokaryotic cell, the only organelle is ribosome, which we are going to learn about that ribosome a little bit detail later on. So from now, we are not going to focus on prokaryotic cells. Those are like bacteria. We are not going to focus because this chapter deals with eukaryotic cell. So when you look at the human body, we have two classes of cells. One group is called sex cells or gametes. The other group is called somatic cells or body cells. The word soma means body. So in case of male, the sex cells is called sperm. Sperm is a plural terminology. Spermatozoa is a singular. And in case of female, the sex cell is commonly known as egg, although it can call ovum or ova, or some book also can call it oocyte. So those are sex cells. Okay. Somatic cells. Somatic cells are the body cells, meaning any other place in your body, liver cells, skin cell, lung cells, anything that is not sperm and oocyte are called somatic cells. So when you study the cell function, we know that it's called cellular physiology. And when you study the cell structure, we call them cytology. Cytology is a study of cell, and histology, by the way, is a study of tissue. So these are the two different things. Histology means group of cells. Like when you're studying a tissue means group of cells, that histology. Cytology is a study of single cell. So that's pretty much the beginning. Now we are going into the structure of the cells and what is outside and inside of the cells. So the fluid outside of the cell, we call it extracellular fluid. Now, whatever it is extracellular, it is also in between multiple cells. So let's say there are 5 cells. So there will be cellular fluid in between those cells. That's why extracellular fluids is also known as interstitial fluid. That means fluid of the tissue. So extracellular fluid and interstitial fluid is the same thing. Inside the cell, which is not mentioned here on the slide, it's called intracellular fluid. So do not get confused between the word interstitial fluid and intracellular fluid. Those are the two different things. Interstitial fluid, which is the tissue fluid, which is also extracellular fluid. That means the fluid outside of a cell. The cell is surrounded by the plasma membrane or cell membrane. So the plasma membrane, their primary job is to give a structure of a cell as well as they keep the liquid or any environment outside of the cell and the inside of the cell separate. So they have separate contents. Inside the cell, the content called cytoplasm, the outside is extracellular fluid. Now inside, in the cytoplasm, in most books, all the organelles will be listed together, but in some book, they put the nucleus a little bit separate. So that is why I put the nucleus right here and the remaining we call cytoplasm, but actually most book can put them together as well. This really does not matter. But here is one more, the word that people often get mistake with, that's cytosol. There is a big difference between cytoplasm and cytosol. Cytoplasm is everything together. Cytosol is the fluid part. Cytosol is the fluid part. So cytosol and the organelle together makes the cytoplasm. Okay. So now we are going to focus on the plasma membrane first, and then we are going to learn the plasma membrane and its role in the membrane transport. Then we are going to go with the organelles in detail. Again, this is the same structure that we just defined earlier. That's the membrane, cell membrane. Inside that is nucleus. Right next to nucleus, the huge membrane-bound structure that's almost all over the place is called rough endoplasmic reticulum. And the one that has not ribosome, like if you go detail of that picture, you will see that red dots are on the top of this. Those red dots are nothing but ribosome. And when the ribosomes are not attached to this area, we call smooth endoplasmic reticulum. Both of them we are going to learn a little bit later as well. Then there is another membrane-bound structure, like a membrane-derived structure. Those are also called Golgi complex or Golgi apparatus. Mitochondria. Then there are lysosomes. These vesicles are called lysosome. Then the other vesicles could call them peroxisome. All these organelles we are going to learn later on. So what is plasma membrane made of? That's very important. And we learn in our biochemistry lesson that there is a molecule called phospholipid. That's the molecule, phospholipid. So I'll use this picture first. This is the molecule phospholipid. If you remember, glycerol, they have 3 alcohol groups. So one alcohol group make that ester bond with a fatty acid. That's another one, another fatty acid. And then, so that's so-called diglyceride. And then instead of putting the third one for making triglyceride, instead they put a phosphate group along with the organic molecule right there. That becomes their head. So that's the head is very polar, meaning they have a lot of charge. It's known as hydrophilic end. And this side is hydrophobic end. This is extremely important for this chapter. So phospholipid is a very interesting molecule. They have 2 parts. One part, the head part, is hydrophilic, meaning they do like water or any, remember water is a polar compound. So they do like water or any other ions. So whoever has charge, they like them. Hydrophobic end, on the other hand, hydrophobic does not like anything that has charge. Does not like that. So hydrophobic tail absolutely do not like anything with charge. So now both of them together, like they have 2 layers of cells. So when scientists discover finally, they came out with this, the model, this is called bilayer model. That means we have 2 layers of phospholipid, one side of the head facing outside of the cell, the other side of the head facing inside of the cell. So inner core, we can use the word core. So inner core of the membrane is all hydrophobic tail. So what does that tell you? That tells you that any ions right here cannot just come here because this part is not going to allow them. Or any ions right there cannot just go there because this part won't allow them. So if it is a small other molecule, yeah, possible, they can move across the membrane, but ions, they can't. But they don't at all? No, they do. There are some other technique. We're going to learn those things. So right now, this is the basic structure. But when you go to the detailed structure, we'll see the plasma membrane is made not only with the phospholipid bilayer, but also a lot of protein molecules. Some protein molecules, they're everywhere. That is why it is known as fluid mosaic model. Like it's a mosaic. So the proteins are like a mosaic, like they are everywhere on the membrane. So some proteins are spanning through the membrane, so they can be both sides. Some protein, they're attached kind of like inside. Some proteins, they can be attached outside. And in this picture, we don't have, but some proteins can be completely embedded inside the membrane. So there are various type of proteins, okay? Some proteins, what they're doing, these proteins are the so-called gate. That means they are the one they're allowing some molecules to move back and forth. Okay, so that's what we are going to learn later on. So these are the proteins which are helpful for movement of the molecule from both sides of the membrane. And sometimes these proteins are also attached to the carbohydrate residues. So these are carbohydrate. So these carbohydrate residues are normally responsible for recognition purposes. So it's like an antenna outside. So they are talking to the other cells. So they recognize each other. Okay. But as the protein become the integral part of the membrane, so if somebody asks you what makes plasma membrane, our answer would be phospholipid bilayer and protein molecules. So plasma membrane is made of mainly phospholipid bilayer and protein molecules. That's their main structure. Now, yes, carbohydrates are attached, but they are not directly applicable. So the carbohydrate attached to the protein or carbohydrate could be attached to the lipid directly. Those are glycolipids. So carbohydrate is this is the example I would give you. So let's say you are looking at a building, like your house. So maybe I would say, okay, the wood and the brick, all these materials, or glass, like normally these days the buildings are like big buildings, not houses. The big buildings are like wood and the brick and the glass. So these are the steel. These are the main thing. But that doesn't mean that paint does not exist. Paint is there. That's for like decorating, designing. Okay. There are some lots of other molecules, other material attached to make a house. Same thing here. So here the carbohydrates are like paints. Okay, they're not essential material, but they are there. And there is another material here. This tiny molecule, this 4-ring structure molecule, those are cholesterol molecules. So cholesterol, particularly in animal cell membrane, they have an interesting function. Basically what happens when the membrane, they want to keep it fluid. That means all the tail, excuse me, all this tail, they're movable. So they're not rigid. So molecules can just go through them. They're like tail, movable tail. Or actually, in reality, those phospholipid can replace their places. So they're movable. Okay, they're dynamic, in other words. But if it is high temperature, the cell become too dynamic, which could damage the cell integrity. So to protect that, these are like kind of like a wedge in between. So they don't want to make it too fluid. On the other hand, when it is a very cold atmosphere, in the real, if you look at the polar bear structure, like they live in a very cold temperature, then you don't want the cell to be too rigid. So the cholesterol molecule, again, they are kind of wedge in between. So they don't want to make the cell too rigid in the cold. They don't want to make the cell too fluid in the high temperature. So we actually some book will call this cholesterol as a temperature buffer. So they are like acting as a temperature buffer, meaning they keep the membrane fluid at most time. Okay? So this is a fluid mosaic model of the membrane. This is the structure of the membrane. But again, essentially the membrane is made with a lipid bilayer. Their head is facing either outside or inside, and protein molecules. Now, based on this structure, we are going to focus on how molecules move. Okay. So molecules movement, which is known as transportation across the membrane, divided into two groups. One group is called passive transport. The other one is called active transport. Okay. So in passive transport, when molecules, so like ions and molecules, they move with their concentration gradient, with their, meaning high to low. So if it is high to low, so with that's gradient, high to low. So they move with their concentration gradient. So they're going to move from high to low, this direction. And they don't require any ATP, or they don't require any energy. Active transport, on the other hand, moving of ions and molecules, movement of ions and molecules against their concentration gradients. That means If they're low, they will move from low to high, that direction. The cell sometimes need it. So they're going to force them to go from low to high. And they do require ATP molecules or the energy molecules. So passive transport is the movement of ions and molecules along with their concentration gradients, does not require energy. Active transport, they move against their concentration gradients and does require energy. Now, passive transport, although they don't require energy, but they could be various types. So just for an example, like we live in this neighborhood. So just for an example, I'll give you an example. Let's say you're walking down. So you're going from one place to another place walking. I would not, so you're not spending money. So I would say, okay, in this case, money is the energy. So the money is the ATP. Or you can take a bicycle go from one to another. You're not literally spending any money, apart from buying the bicycle. That I'm going to assume that you have it. On the other hand, you're taking the train, you're taking the bus. Those are like takes money directly. So those can be considered as active transport. This is passive transport. I'll give another example just to understand it. Let's say you're driving, you're going through in the city, and you're going through Midtown Tunnel from Queens to Manhattan, and they charge you toll. So that's active transport because you need money. And if you go through Queensboro Bridge or Brooklyn Bridge, there is no toll. That means, let's say, that's a passive transport. You can think that way. Okay. So passive transport does not require money. Active transport does require money. The money means, in this case, ATP molecule, the energy molecule. Now, passive transport can be multiple types. So what are the types of passive transport? Passive transport, I'm going to come back to this particular picture, 2 seconds. This is the type. So they could be called simple diffusion, or just commonly known as diffusion, facilitated diffusion, diffusion with some facility, osmosis, filtration, and dialysis. These 5 types are passive transport. So one thing is common in all the 5, that they don't require any ATP, and they do move, the molecules do move from along with their concentration gradient, meaning from high to low concentration. Okay, but in this chapter, we're going to focus only these first 3. So we're going to mainly focus on simple diffusion, facilitated diffusion, and osmosis. These 2, although passive transport, we are going to mainly use them in ANP 2. So let's go back right here. So this is a simple explanation of the passive transport, those 3 that we should learn: diffusion, facilitated diffusion, and osmosis. So in one simple way, how it is defined? All of them, in all the cases, the molecules are high concentration here. They're moving to wherever the concentration of the molecules is low. Okay, now, excuse me. Active transport, molecule concentration is low, where they're moving towards a molecule concentration is high. Now, diffusion, they're simply moving through the membrane. Facilitated diffusion, they're also moving from high to low with the help of a protein molecules. This background, that molecule is a protein molecule. And in this case, this is very interesting. Lower solute concentration, meaning high water concentration. And osmosis is the movement of water. So although in this picture, this is how people get confused. In this picture, they're showing, okay, only 3 molecules here, there's so many molecules here, so high concentration here. This is true. But osmosis is not movement of the molecules. Molecules are not moving. This molecules means solute are not moving. Who is moving? Water is moving. So water goes, if there is, I'll give an example. So let's say it's a 3% solution, and another case, let's say a 9% solution. Okay, so 3% solution. The reason I said 3, because there are 3 green dot indicating like 3 molecules here, and right here 1, 2, 3, 4, 5, 6, 7, and 9, 10. Okay, so let's say 10% solution on the other side. So what will happen is, if there are 3% solution, that means 97% is water. If there is 10% solution, that means 90% is water. This is key. This is very important. So when you have a 3% solution in a cup, let's say there is a cup called 3% solution, another cup has 10% solution. The 3% solution is lower concentration. True. The 10% solution is a higher concentration. Now, if I join a pipeline between those two cups and I don't allow the solute to move, I allow only water to move. So now water is moving. Now the question is which direction water will move. Although 3% is low, 10% is high, they're not going to move from 10 to 3 because, as I said, solute is not moving. Water is the one moving. So when there is 3% solution, in back of our mind, we know that 97% is water. 10% solution, meaning 10% solute, 90% is the water. So 97 obviously higher than 90. So the water is going to move from 97% to 90%. Although we don't say that water concentration, but in this case, we're going to follow that. So where the lower solute concentration, water is high. When there is a higher solute concentration, water is low. And remember, osmosis is technically diffusion of water. Osmosis is in quotation like diffusion of water. So where water moves from high water concentration to low water concentration, but the word we don't really use in science, something called water concentration. We call solute concentration. So when we say concentration, we refer to the solute. But in this case, in our mind, we're going to remember water is the one that is moving. So that's passive transport, no energy required. Active transport, they're moving from lower to higher solute concentration, so against the concentration gradient, and they require ATP. They also require protein molecules which help them to move, and there are more detail coming up later on. So let's study the diffusion first. So what is diffusion? So let's understand this. If I take a room freshener, so if I take a room freshener and let's say I'm sitting in a room, so I'm spraying here a little bit room freshener. After 5 minutes, if I go far away at the other corner of the room, I will be able to smell them. The question is why? The answer is the molecules that we just sprayed right here is high concentration here, and they're going to move slowly. So which direction they will move? They're going to move in all the directions. So they're going to be moving in a circle. So they're going to be moving in all the direction wherever they're low. So high to low. So in this example, they put a crystal right on the bottom of the cylinder, the let's say colored dye, and after some time it will look this. Why? Because high concentration of dye, now the molecules started moving slowly. So after even some more time, they're going to be dispersed everywhere. That means they're equilibrium. So those molecules or ions are going to move from area of a high concentration to area of a low concentration, and they're going to move in all directions until they reach an equilibrium. Another example I'll give you, like, if you buy a soda can of soda, and as soon as you pop that soda can, you see the hissing noise comes out because carbon dioxide in high pressure in that soda can. As soon as you open that carbon dioxide comes out instantaneously a lot, and then slowly can keep coming out because the universe is too big. So it's almost so-called all the carbon dioxide molecule will come out, and we call them like flat after some time. So the soda will become so-called flat after some time because the soda will release almost all their carbon dioxide, because compared to the soda can, as I said, the universe is too big, so the molecule will keep getting dispersed. Same thing, diffusion. So diffusion is basically movement of molecule from wherever they are high concentration to low concentration. Now, are there factors that can affect them? Of course. The diffusion is, first of all, they're affected by the concentration gradient. That's the most important thing. So higher the gradient, meaning higher the steep, faster is the movement. So if the concentration gradient is like this, so there will be a little movement from high to low. But if the concentration gradient like that, so if it is like 10% to 9%, yes, they will move. But if it's 10% to 1%, they will move faster. So steeper the concentration gradient, faster is the movement, faster is the diffusion. And on the same way, think about distance. So between two cells, if the distance is too small, the diffusion will reach faster equilibrium. So for example, instead of in the cylinder, if you don't have that much water, let's say you have this much water, the diffusion will be for less time because they are going to reach equilibrium quickly. So more water means they will take longer time. Okay? Because if the shorter is the distance, they eliminate the concentration gradient too quickly. Then there is a direct effect on the diffusion rate is the temperature. Temperature will directly affect the higher the temperature, faster is the movement. Higher the temperature, faster is the movement. Then size of the molecules is extremely important. Size of the molecule. So bigger the size, larger the molecule, slower is the movement. So if you have two molecules, one is, let's say, the X molecular weight, the other one is 2X molecular weight. Obviously, the one with the 2X molecular weight will be slower. How slow it is? Half the time. So if X can move 1 centimeter, by that time, 2X will move half a centimeter because they are double the molecular weight. Okay. So diffusion can be directly affected by concentration gradient, distance, size, temperature. And there is another tiny factor when you're talking about the ions, because there is a factor called electrical force, meaning not only now the concentration gradient, but also just imagine this. Inside the membrane, inside the cell, we have more negative environment. In a cell, we are going to learn that detail in nervous system. So without going into detail, if this is a cell, inside the cell, we have more negative environment. So when the molecule, negative environment, so now if the ions are positive, and if they have a concentration gradient from outside to inside, they are now moving from outside to inside in any particular situation, they're going to move a little bit faster because they want to be attracted towards the negative direction. But if you're talking about, let's say, another negative ion, let's say chloride ion, although there is a concentration gradient, but because the negative repel other negative, the movement will be slightly slow. So we can say that electrical force can also affect the movement of ions. Okay. Now, keeping those everything in mind, when the diffusion takes place through a human cell, these are the two things very important. Yes, concentration gradient is important, but these are the two things very important: size and the charge. So if the molecule is big, they can't really move because through that phospholipid bilayer and a lot of protein, they can't really move. If the molecule, if it is not a neutral molecule, let's say it's an ion, let's say sodium ion, potassium ion, chloride ion, magnesium ion, they really cannot move because remember, phospholipid bilayer, that inside hydrophobic core, they don't allow ions to move. So when it comes to the human cell, the easiest example, when you breathe in oxygen, oxygen can diffuse into the blood. From the blood, carbon dioxide diffuse back and you breathe out. So oxygen and carbon dioxide can diffuse back and forth, not a problem. But sodium, potassium, chloride, these can't. Or even large molecule, let's say a large amino acid, large glucose molecule, they can't because they're large. So when they're large but essential, such as glucose and amino acid, they take another route. It's called facilitated diffusion. They take another route. It's called facilitated diffusion. What osmosis, I'm coming back. Let me just talk about this first. So facilitated diffusion, what this is very, think about, I'll give an example to understand. Let's say you are in town A and you want to go to a town B or whatever it is. Like you're in school, you want to go home. Okay, you don't want to walk. You do not want to spend money. And the question is how you're going to go. The only way you can go is somebody give you a ride. So somebody give you a ride. So that person is paying for everything. You would just be nice with that person. So you're getting a facility from that particular individual. That's called facilitated diffusion. So in our body, cell does the same thing. They have molecules sitting there. The molecules sitting there, they are not charging money, meaning there is no ATP involved. So high amount of glucose, they're just taking them in. These molecules are literally called glucose transporter or GLUT, glucose transporter or GLUT. So basically what they do, they help those proteins are integral part of the membrane. So they are sitting on the membrane. So these molecules are giving the facility so that glucose or amino acid can diffuse across them. So when they diffuse across them, diffusion meaning high concentration to low, but with the help of these molecules, we call them facilitated diffusion. Very important, particularly these two molecules you should know, glucose and an amino acid follow this route. Then comes osmosis. Now osmosis, as I explained earlier, the osmosis is the diffusion of water. So what is diffusion again? Diffusion is the movement of molecules from high to low concentration. So osmosis is diffusion of water. So movement of water from high water concentration to an area where low water concentration. Although we do not use that term, I wrote down and underlined just for your understanding. So we do not call water concentration, but we understand what water concentration means, meaning low solute, higher water. Higher solute, less water. You have to keep that in mind. So now let's imagine three different situations, three different scenarios. So you're talking about a cell. So this example, we're talking about a red blood cell, which looks like a donut, like a all perfectly circle. Like when you see under the microscope, a red blood cell appear a perfect circle. Okay, so that's why this picture is like that. So red blood cell, let me explain first. Inside the red blood cell, the ionic environment is equivalent to 0.85% of sodium chloride. Okay. So at this point, I want you to write this down and look into the book later on as well. It is 0.85%. In some book will round it, it says 0.9%. So inside the red blood cell, the ionic environment is equivalent to let's say 9%, so 0.9% sodium chloride solution. So what does that mean to your head automatically? If 0.9% is the solute, that means 99.1% is water inside the cell. So your inside the cell environment is 0.9% solute, 99.1% water. If outside you have the same 0.9% solute, that means it's both sides is same. That kind of condition we call isotonic condition. Tonicity means, they use the word as how, depending on how much solute you have. So iso means same. So isotonic solution means both sides you have the same solute concentration, 0.9, 0.9. The reason we're using the word concentration 0.9 is known as saline. Okay, I'm going to mention about that 2 seconds later. Another situation, hypotonic, meaning inside we know that 0.9. Outside, we can be, let's say, pure water, meaning 0% solid, or anything lower than 0.9%. Anything lower than 0.9%. And hypertonic means outside should be higher than 0.9%. So let's say 1.5% or 10%, higher than that. So what happened in these three situations? So what happened in isotonic, there is no concentration gradient because inside water and outside water is same. Water is not going to move, meaning there will be no significant movement of water. If one molecule go outside, the other molecule will come inside. That's why it says no net movement of water. Yeah, water is moving, but there is no net movement. If 10 leaves, 10 comes in. So no net movement of water. So the cell remain exactly the same, the size of the cell. In hypotonic, outside is lower than that of the cell, solute concentration. That means outside has more water. This is the part you have to understand. If the outside is a lower solute concentration, that means outside is more water. Inside, remember 0.9, so 99.1% is water. Let's imagine outside is 0% solute. That means 100% is water. So 100 is obviously higher than 99.9. So the water will move from outside to inside. So in a hypotonic condition, water moves inside. So what will happen to the cell? Cell will swell and then eventually burst because we don't have cell wall. So there will be nobody to protect us. That bursting of RBC, we call it hemolysis. Lysis means breaking down. Okay. In hypertonic, opposite happens. So the cell lose water. So the water goes from inside to the cell to the outside. So when water goes out, the cells shrink. That phenomena is called crenation. Crenate means shrinking or wrinkling. So cells shrink in the hypertonic condition, cells swell and burst in the hypotonic condition, and isotonic, no change. That is why when you are in any emergency situation, or a lot of people, when they're dehydrated, doctor inject them or they give them IV with saline. Saline is 0.9% sodium chloride. That means they are giving you isotonic. They cannot give you plain water. If they give you water, you will die in half an hour because all your RBC will burst. They cannot give you a higher concentration of solute because then your cell will lose, it will get dehydrated. So they will have to give you saline where they're putting a lot of water in your body, but your RBC is not disturbed. That's osmosis. Okay, just to mention, just to understand here, in hypotonic condition, plants love that. That's why when you do not water your plant will wilt. And as soon as you water, they stand straight, because hypotonic, you put water, water goes in, the cells swell, and when they swell, they stand erect because they have cell wall, so they're not bursting. Okay? Just on a side note. So this is extremely important. Make sure you go over them a couple of times to understand exactly how osmosis works. So that's together is diffusion, osmosis, and facilitated diffusion together is the passive transport. Again, you should know that dialysis and filtration also passive transport, detail not required at this moment. Active transport. So active transport is the movement of molecules or ions against their concentration gradient, and obviously ATP is required. Okay, so there are various kind of active transport. One first type is called protein pump. These are all transporter proteins exactly located like the glucose transporter, but glucose transporter does not cause ATP. So that's a facilitated diffusion. This one is asking for ATP. So in moving the molecule, they're asking for ATP. So these are called protein pump. In your body, in the cells, you have various kind of protein pump, like sodium, potassium exchange pump, or sodium potassium ATPase pump. It's called ATPase because it's using ATP. There are hundreds and thousands of them. They are continuously taking sodium from one side. There could be hydrogen pump, or simply known as proton pump. There could be calcium pump, magnesium pump, zinc pump, and so on. Okay, so for every single ion, we have a pump. So I'll give an example to understand this one, because you know this. Proton pump. Proton is, remember, the proton donor is the acid. So in your stomach, you have tons of proton pumps. Okay, why? Because they're secreting proton into the proton means hydrogen. Proton pump into the stomach. So stomach is very acidic. Now, some people you suffer from, like you, I'm sure, heard about, it's called acid reflux disease, meaning they secrete too much acid, so they give a lot of problem in this area and burning sensation and so on and so forth. So the medication you take nowadays, the third generation medication, not just simply alkali. So you take something called omeprazole or something called the purple pill, like the Prevacid. Now, what are those? So those are nothing but, if you look at on the bottom of the medicine, it says proton pump inhibitor, or PPI drug. So proton pump inhibitor means they go and inhibit that pump. Inhibit means stop that pump. So when the proton pump is not working, they cannot secrete proton. And if they cannot secrete proton, you cannot have that much acidity. That's the understanding. So this is how actually scientists find out the cure for the disease. Okay, so that's one active transport. Then the second type of active transport is called vesicular transport. Vesicular, meaning they create a vesicle, the container. Okay, so vesicular transport, there are three varieties. I'm first going to explain the first two, endocytosis and exocytosis. Okay, the third one is receptor-mediated endocytosis. So some book will put them in the endocytosis area, some book put them a little bit separate. Okay, So let's learn three of them. So endo and exocytosis. Endo means taking in, exo means throwing out. So basically this is what you have to do. Is cell exactly like us. Think about if I have to eat, what do you have to do first? I have to do first. What I have to do first is I have to open my mouth like this. So you open your mouth and then engulf the food and take it. Cell does exactly the same thing. Their plasma membrane, as I said, they are flexible. So they go around the cell and they literally open and engulf it. That's endocytosis. Exocytosis, same thing. You open your mouth eventually and throw things out. So exocytosis. So if you look at this picture right here, the top one is exocytosis, meaning let's say these are the waste product you want to throw out. So this is the inside of the cell. This is the membrane-bound vesicle. So that's a vesicle containing the waste. They come close to the membrane, they become the part of the membrane, and look at what the molecules are outside, throwing out. So you're spitting out, exocytosis. Endocytosis, here are the molecules, you want to take them in. So how to take them in? They get folded. It's like exactly you're opening your mouth and then you engulf it. So open your mouth, take the molecule inside, engulf it, boom, inside. Now, when you're taking inside, there are two things. Say for example, I'll give you a bowl of salad. If I give you a bowl of salad and like lettuce, tomatoes, etc. So I say, hey, just eat the tomatoes. And I think you can do it. You can take a fork and just, take the tomatoes and you can eat it. So you're taking in only tomatoes. You have a choice. Now, if I give you a glass of milk and I said, hey, just drink the calcium. Now, not possible, because now in order to take the calcium, we know the milk contains calcium, but in order to take the calcium, you have to drink the whole thing. So the endocytosis is two types. If you pick and choose, meaning you're eating, phagocytosis, and if you're drinking, like you drank the milk, pinocytosis. So when you're looking at one virus or one bacteria, the cell engulf them and kill them, that's phagocytosis. And pinocytosis is this one, like a bulk transport. This is called bulk phase endocytosis, meaning they're taking everything in, liquid. So when they take liquid, they have no choice. So endocytosis in that regard, two types, phagocytosis and pinocytosis. Then there is a third variety of endocytosis, which is called receptor-mediated endocytosis. Nowadays, we put them completely separate. So what that means look carefully. So this is the cell. So this is the inside of the cell, that's outside of the cell. In the outside, there are a lot of target molecule. And this molecule, sometimes also known as ligand. That is why some book will call it, instead of receptor-mediated endocytosis, they can call ligand-bound endocytosis. So this ligand has to bind to the receptors. So if we do not have those receptors in this picture, the blue color material, if they don't have the receptors, they can't take them in because they don't identify them. So in other words, you have to identify them, bind them, then take them in. So receptor bind them, the molecules is bound, and then they take it in. And once they take it in, lysosome, break these things, break these vesicles, the lysosome fuse with them, they take out the molecules they need, and the receptors goes back to the plasma membrane again. So this type of movement, we call it receptor-mediated endocytosis. Cholesterol is one of the, cholesterol is the one that goes like this way. Okay? So if you do not have enough receptors on your body, you may have higher cholesterol. So that's why some people genetically have higher cholesterol in their blood, called hypercholesterolemia because of this. So that's vesicular transport. So that is all the transport and the plasma membrane you need to know. This is the most important part of the chapter. Important in the sense this is the part that you need to understand. The second half of the chapter is basically all the organelles, and you need to know what they look, how they function. So I'm going to go through some slides, but this is where you come in. So this is where your study habit comes in. So I would recommend you have to write down all these things whenever you get a chance, like all the organelles and what they do. All the organelles and what they do. That's what basically you have to do. So starting with the ribosomes. Ribosome is a small package like it appears like in this laser pointer, like a tiny red dot in my picture. So let's say, look at the picture. These tiny red dots, those are ribosomes. In this case, like a pink dot, those are ribosomes. So ribosomes are a package of protein and RNA molecule. Okay? Ribosomal RNA and protein combined together, but they do an extremely important job. They're responsible for protein synthesis. They're responsible for protein synthesis. So protein, how they make? From DNA, we make RNA. From RNA, we use that RNA as a code, and ribosome translate them into chain of amino acid called protein. That's the central dogma of biology. So without ribosome, a cell will not be able to make protein. So if a cell is lacking ribosome, they can't make protein. Now ribosome could be either free or either membrane bound. So if they're free, we call it just they're roaming around in the cytosol, we call it free ribosome. They can also synthesize protein, but they're not so efficient as the bound one, but they can still synthesize. Those proteins are normally utilized within the cell. But when the ribosome are sitting on endoplasmic reticulum, so they're bound to endoplasmic reticulum, that portion of the endoplasmic reticulum, we call them rough endoplasmic reticulum because they appear rough because all the ribosomes are sitting on them. So endoplasmic reticulum, the humongous organelle outside the outside the nucleus, is two types: rough endoplasmic reticulum, smooth endoplasmic reticulum, based on the fact where ribosome is attached. So ribosome is attached to the rough endoplasmic reticulum, and they also make a lot of protein. Those protein normally gets modified and get transported. So either they get transported to the plasma membrane, so they make the plasma membrane protein, or they can go outside of the cell. The ribosome, their job is to make protein. Now, endoplasmic reticulum, as I just said, there are two types: rough ER and smooth ER. Rough ER means where ribosomes are attached. Smooth ER means ribosomes are not attached. Both has individual lot of functions. So the rough ER does, ribosome, rough ER, and Golgi, they have a sequential job. So I'll use this particular picture to explain. So these are the ribosomes. They're sitting, this huge membrane is rough ER. So they're sitting on the rough ER, they're making the protein. So the protein is made right here. Now we already learned that protein undergo modification: primary, secondary, tertiary, quaternary. All this modification takes place here, and then they can create a vesicle. And that vesicle goes to Golgi apparatus. This is Golgi apparatus, another membranous structure. Golgi apparatus, their job, I mentioned later on, is like, let's say, FedEx or UPS. So what they do, they receive the package, they modify them, and then they ship them. So that is why this part of the Golgi apparatus is sometimes known as receiving face. So that's the entry phase. Some is called receiving phase. So they receive the protein, then they modify them more. Sometimes protein may need to attach with the carbohydrate residue. Sometimes protein required to clip up something to make more. Those are like higher level study. You're going to learn protein modification. So protein modification can take place there, and then they make a vesicles. So either they can make storage vesicle, or they can make vesicle that goes towards the membrane, or they can make vesicle that goes towards the secretion. So when they goes to the membrane, that becomes part of the plasma membrane. That means plasma membrane is synthesized that way, like all the protein in the membrane. And sometimes they can secretory. Secretory means they are going to release, like hormones or any other protein that protein a cell you want to secrete. That goes here. And also another part of this to create the lysosome, meaning all the enzymes, they put them and they create lysosome, which is another organ. So this is very important. This is how the protein passage from the endoplasmic reticulum to Golgi apparatus to the plasma membrane. This is all their function explained. So this is how ribosome, when they're bound with ER, they do their work. Smooth ER on the other, that's why I wrote down for Golgi, that's why I wrote down FedEx, by the way. FedEx has nothing to do with Golgi. Golgi is somebody's name. Okay. Smooth ER, on the other hand, they have distinctive functions, meaning they are responsible for synthesizing phospholipids, cholesterol, steroid hormones, such as like, steroid means cholesterol for the membrane, and then estrogen, progesterone, all the steroidal hormones. And they can also make carbohydrate. So this is lipid. I forgot to mention here, carbohydrate makes sure they're also responsible for making glycogen in the liver cells or in the muscle cells. So they help in synthesizing different type of lipid. They help in synthesizing different type of carbohydrate. And then they have two additional jobs. One is they detoxify harmful substances such as alcohol, particularly in the liver. In hepatocytes, they're well-developed smooth ER. They always help us to these harmful substances, detoxify them. And then finally, smooth ER, which is not mentioned on the slide, but make sure you study this. Finally, smooth ER also helps storing calcium ions. For example, smooth ER in muscle cell, they get modified. We are going to learn in muscle cell in detail what they do. They store calcium ions, so they can act as a storage as well. Okay, so smooth ER compared to the rough ER has a distinctive function. They don't have to do anything with the protein, but they do lipid, carbohydrate, detoxify substances, and storage of calcium. So we now know ribosome, rough ER, Golgi apparatus, and smooth ER. So I'm going to skip this. I'm going to come to lysosome. So lysosome, again, like zome means packet. So lysosome is a packet full of lytic enzymes. Lytic enzymes, meaning they're digestive enzymes. They're called hydrolytic enzymes. So they use water to break everything. So anything that goes in your cell, lysosome will attack them first. Anything that goes into the cell, lysosome will attack them first. Then they will chop them down. Now, if it is bacteria, you are good because they save you. And if it is sugar or any other energy molecule, they're going to produce energy. Okay, so that's their job. So they digest any foreign substances that gets into your cell. Now, sometimes it's called autophagy, meaning what they do, they recycle the damaged organelles. So let's say mitochondria is not working. One of the mitochondria is not working for some reason. So cell is not going to keep that. Cell will recycle that. Who does that? Lysosome. So lysosome will engulf the mitochondria. They will combine them. They will chop them, digest them, and recycle the products. That's why it's called autophagy. Phagocytosis. Phagy means eating. Autophagy. They eat their own. And that's exactly what happened when an organism dies. So let's say I die and nobody moves me, and I'm keeping my dead body in this room, and after a year, there will be nothing. Why? Because autolysis, meaning my cell will burst open, the lysosomal enzyme will come outside, and they will recycle the whole product. This is the picture they were showing, like if the food goes or the mitochondria, damaged mitochondria, lysosome is digesting them. Peroxisome, very similar, similar in the sense there is also a lot of breaking down going on, but it's totally different compared to lysosome. They're slightly smaller. Lysosome generated from Golgi apparatus. So they are in the same membranous system. Peroxisome is different. Peroxisome, they can develop their own. Peroxisome is one of the organelles like mitochondria. They can grow themselves, so they can divide to make more in the cell if needed. So what they do, their main job is to, they also deal with toxic substances such as alcohol and formaldehyde. They also deal with like fatty acid and other organic molecule breakdown. But here's the important thing. When they break down, they take a route different than the other because they break down by production of hydrogen peroxide, H2O2. That is why it is called peroxisome. Now, why this is important? Because hydrogen peroxide itself is dangerous. So why they are doing that, like why they're breaking down in a route when they produce a dangerous waste product, like a toxic waste product? Because they have a special enzyme. They have special enzyme. They can remove hydrogen peroxide, meaning they can convert hydrogen peroxide to water. Those are like a catalase, for example, one of those enzymes. So they are very specialized organelles in that regard. So they deals with fatty acid, they deals with other organic molecules, they deals with alcohol, but they take the route of production of hydrogen peroxide. That's why the name peroxisome. Mitochondria, sometimes also known as simply powerhouse of the cell, meaning because they are responsible for generating most ATP, not all, most ATP in the cell. Mitochondria, as I mentioned, like peroxisome, they are self-replicating, meaning they can make their own more mitochondria if they want, okay, when the body demands more ATP. Their double membrane structure, very important to know mitochondrial membrane, because there will be a chapter called cellular respiration. We're going to need that. They have a double membrane structure. This is outer membrane, in this picture, red color. The yellow color membrane is the inner mitochondrial membrane, which creates a lot of folds just to increase the surface area. So if you follow the membrane, they create a lot of folds. Those folds increasing the surface area. Those folds are called cristae. So cristae is the internal membrane fold. And why there is fold? Because they want to increase the surface area. Why they want to increase the surface area? Because they can fit more enzyme for cellular respiration, which we are going to learn later on. So right now, you should know their structure. So later on, it will help you to understand its function better. And inside of the inner membrane, the area called mitochondrial matrix. In the mitochondrial matrix, we have mitochondrial DNA. Whether you know it or not, interestingly, DNA is not only in the nucleus. Okay, mitochondria, they do have their own DNA, they do have their own ribosome, because they can make their own protein. That's why some ribosomes are inside the mitochondria. They can make their own protein. And now we have this interesting fact. I don't know whether you know it or not. All your mitochondria, you got it from your mother. All your mitochondria, you got it from your mother, because that's what the egg is. Sperm only delivers the half of the chromosome material. So mitochondria, the DNA, if you look, it will be 100% matched with your mother. Okay. Cytoskeleton, it's like our skeleton system. So the cell skeletal system. So it's like a bone structure inside. Basically, those are all protein molecules. So they create like the beams, the rod, they create, they keep the shape and a lot of other functions, okay? There are three varieties based on like microtubules, intermediate filaments, and microfilament based on how thick they are. So thickest one called microtubule, thinnest one is called microfilament, and in the middle we call intermediate filament. Individually, they have different protein, they make it. Individually, they have different function, and you should know it. So microtubule, let's study with this one first. Microtubule, the thickest one, made with a protein called tubulin. That's the protein they make it. So they help you, they act like a monorail system. What does that mean? Then just imagine the cell for a protein or for another organelle, the cell is like New York City, is big. So the tubulin, they sit down like they're like everywhere in the cell. So they're like a monorail system where, let's say, a mitochondria can hook onto the tubulin and glide from one place to another place. That's what they're saying. Basically, in other words, microtubule help in transportation inside of a cell. Microtubule helps in the transportation of molecules inside of a cell. When you study nervous system, this will come into the very help. They also form spindle apparatus or spindle fiber, which helps into your cell division. So in the cell divide, those strings that get attached to the chromosome, I think you saw those structures in biology to make the chromosome goes on the two corner of the cell. Those are called spindle. They are made from microtubules, okay? Then obviously there are cilia and flagella, which we'll learn later on. They also made with microtubules. Intermediate filaments, they're made with a protein called keratin. That is the same protein that make the hair. So very strong protein. They have also different type of function, particularly one in your skin cell. The keratin makes it very strong. And microfilament, the protein called actin, there are many functions. So one of the function is that actin involved with myosin for produce muscle contraction, or other words, even cell movement. Muscle contraction we're going to learn later on, cell movement. And also the microfilament has another function. When the cell division takes place, the cell divide into two. In biology, you learn that in the middle, that called cleavage formation. That cleavage is done by actin, okay? Again, these are the simplest function that I listed here. Please read the book first and then this, in order to understand which protein makes what and what are their functions. Then comes flagella and cilia. Flagella and cilia. Flagella and cilia both are extension of the plasma membrane. Both are extension of the plasma membrane. And inside them, they have microtubules to help them move. Okay. Now, flagella is like a tail. So they help the cell to move. And only one human cell has it, that's sperm cell. And cilia is the extension of a cell. So let's say these are the cilia. These are the cilia, hair-like structure. So they increase the surface area of the cell. And not only that, they also help moving. Let's say this is cilia, so they help moving the particle that sits on the cell. So right here, your windpipe or the trachea, inside all the cells have cilia. So when you choke by mistake, like food particle goes to your trachea, you choke, you cough. So those cilia are bidding the food particles from that place to back into the mouth. So that's the function of the cilia. So cilia moving the particle that is on them, and flagella moving the cell itself. So this is how they bid. So cilia, those are like they're bidding on the membrane so that the molecules can move, and the flagella, their bidding can make the cell to move from one to another locations. And finally, the nucleus. So nucleus, basically, this is the easiest part to understand. The nucleus, if somebody says, what is the function of nucleus? In one word, everything. That's the control center of the cell, okay? Normally, cells have one nucleus, but there are cells in human body, skeletal muscle cell, there are multiple nuclei, and there are some cells in human body, so a red blood cell, they have no nucleus. The mature red blood cell, they eject their nucleus. So there are some cells with no nucleus, there are some cells with multiple nuclei. Most cells, we have one nucleus per cell. What they contain the genetic material. So all the genetic material, which is commonly known as chromatin material or chromosomes, are located in the nucleus, which are made with DNA molecules. Now, what are they? They are your genetic information. So from there, DNA makes the mRNA, messenger RNA, which comes outside because DNA is too big to come outside from the nucleus. So RNA comes outside, and then they make protein using the help of ribosome, which we just learned. Okay. So DNA is the basic, all the information that's stored, and protein do the work. So if you do not have a correct information, you cannot make correct protein, you cannot do the correct work. So remember, most of the enzymes are protein. So if you do not have enzymes, you can't do the job. So if you do not have the correct DNA sequence, you don't have the correct protein, you cannot do the job. So DNA, in other words, is everything. Okay, so the inside the nucleus is a double membrane structure. A little bit about their structure is a double membrane structure, and they have pores. These pores are also lined with other proteins. So the molecules, those are smaller, not the DNA. RNA can come outside of the pore, or ribosome is actually made here. Okay, nucleolus is a darker bodies inside the nucleus. That's what the ribosome synthesizes. So ribosome make here, assemble there actually, the protein and the RNA assemble there, and then it comes out to the pores outside in the cytoplasm. So nucleolus, that's what the ribosome assembly done. The whole chromosome now looks like a silky material inside. Those are chromatin material. During the cell division, they become thick. We call them chromosome. In normal human body, cells have 46 chromosomes, or we call them 23 pairs. More specifically, 22 autosomal pairs and 2 sex chromosomes. So 22 pairs, 44, and then 2 sex chromosomes. In case of a male, it is 1 X chromosome and 1 Y chromosome. In case of a female, it is both are X chromosomes. Together, 46 chromosomes for a normal nucleus in the human cell. Okay, so that's the entire chapter. That's the entire chapter. So when you study the chapter, make sure first you study the little bit information about the cell, whether eukaryotic cell, prokaryotic cell, what are the difference between them. And we are human are eukaryotic cells. We have 2 types of cells, sex cells or gametes, and somatic cells. And then we come into the cell. The first part we discuss about the plasma membrane, and their structure and how it influences the movement of the molecules. So there are two kinds of movements: passive transport, active transport. Passive is various types. Mainly we focus on diffusion, facilitated diffusion, and osmosis. Active transport, we focused on the protein pump. Then vesicular transport. Vesicular transport, multiple types. We focus on endocytosis, exocytosis, and receptor-mediated endocytosis. Then endocytosis is two types: phagocytosis and pinocytosis. So once you learn all the transportation, then you go. But in the passive transport, do not forget go into the detail, like diffusion, a little bit of detail, osmosis. Remember isotonic solution, hypotonic solution, hypertonic solution. What happens in each case? Very important. Facilitated diffusion. Some examples I gave you: glucose, amino acid, how they go from one side to the cell. Do they follow concentration gradient, or are they against all those things? And then comes all the organelles. I gave you a list of organelles. Your book may have additional organelles. Do not need to study. Follow my list. Only these organelles and their functions you must know. So ribosome, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus or Golgi bodies, lysosome, peroxisome, mitochondria. Then we study cytoskeleton, cilia and flagella, nucleus. That's the chapter. Okay. Thank you.